one of our major programmatic initiatives over the last 3 years, which we started in partnership with the National Center for Advancing Translational Sciences at NIH, has focused on drug development and drug repurposing. The goal of which was to essentially survey, if you will, assess the entire library of approved drugs in the United States, Europe, and Japan, as well as thousands of compounds that are on the shelf or in the libraries but unapproved, to look for efficacy in treating juvenile dermatomyositis. This program has been under the leadership of Dr. Travis Kinder, and as I mentioned, it is a part of the National Center for Advancing Translational Sciences at NIH. And what's important to note in that name is that the mission of NCATS, or the National Center for Advancing Translational Sciences, is to do exactly what they are doing for us, to identify drugs in a translational sense that can move from the laboratory to use in actual patients. And this is our goal in this project. Dr. Kinder has some, I think, very exciting news to share, has just published his first paper that details the outcomes of his work in the lab over the last 3 years.
And thank you everybody for your continued support and for everything CureJM has been doing for patients and families and funding our research. As you can imagine, NIH is spread pretty thin. They cover all kinds of human health conditions, particularly now in the coronavirus. I'm sure everyone has seen Dr. Fauci from NIAID on the news a lot. So NCATS is essentially the newest center of the NIH. So the NIH has multiple centers. Obviously NIAID focuses on infectious diseases. NCATS, their mission, as Jim pointed out, is translational sciences. And translational sciences is essentially taking basic science observations, things that scientists have been researching for decades now, and actually turning it into a therapy. So no longer just kind of toiling in a lab, just out of curiosity, but actually creating technologies and therapies out of it. One of our particular areas of interest are rare diseases, because as you can imagine, if we're doing therapeutic development, pharmaceutical companies are heavily doing therapeutic development. However, rare diseases are often neglected, particularly because it's such a small patient population. It's not quite as profitable for them. So that's kind of where our role comes in the early stage translation. So we can take projects from a very early stage that are considered very risky, and we can do this upfront kind of de-risking of projects and come up with leads and come up with some kind of compounds or drugs that might be effective that can then be taken on further by pharmaceutical companies and clinical trials and things like that. I know times are tough right now for everybody, particularly in autoimmune conditions in the JM field. And I want you guys to know that we're still working for you guys. My time, 100% of my time is funded by CureJM. So I've still been nonstop doing CureJM stuff, even during quarantine. So actually everybody at NIH has been staying at home unless you're doing COVID research, obviously. So I had been at home last couple months, and in these last couple months, I published a couple of papers, both on myositis research, one of them from grad school, work I did in grad school at GW, and another one detailing the work I've done at NCATS with the CureJM funding. And so we've been doing this project over the last few years with the CureJM funding, and that's essentially kind of what I wanted to update you all about and the progress we've made there. So we essentially started at ground zero with this project when I came in. And so the kind of paradigm and pipeline for translational sciences, especially in the early stage, is to first get together with experts in the field, which we did. CureJam has a medical advisory board who's very instrumental in this. A lot of the key experts from around the world. We kind of discussed what are the different problems going on in myositis. Like there's too much of this inflammation or not enough of this other thing. And then from there, we can try and develop tests, which we call assays. So an assay essentially has the root of assessment. So an assay is just essentially a test we can do for some of these key components of myositis that we know to be problematic. So for example, in JM, there's too much inflammation, obviously. There's a key inflammatory component called type 1 interferon. And so we've developed assays or tests for this type 1 interferon, this inflammatory component. So what we did was we essentially developed a model of myositis inflammation in a teeny tiny test tube. And these teeny tiny test tubes are arrayed on a small plate. The plates are about the size of a postcard, and they contain 1,536 of these tiny test tubes. We call them 1536 well plates. And so the wells are the little test tubes. And so we put muscle cells into these wells, add the interferon, so it creates a muscle inflammation environment. And then we add, with robotics, we add potentially up to thousands, tens of thousands, or even hundreds of thousands of different drugs or compounds that are kind of in the development process, becoming drugs. And then we can use these tests to see which ones, which of these drugs or compounds can inhibit the inflammation in the dish. And so that's what we've done this early stage. We developed these assays. We did screening to, up to today, we've screened 15,000 compounds, including all approved drugs approved by the FDA in the US, throughout Europe and Japan. And that's what we published on. And we identified a handful of actually very promising compounds. There's actually about 67 compounds that were active. So that's a lot. And so the next kind of crucial stage is kind of whittling down and prioritizing which are more close to being translational and which ones are farther down the road. So some of you have maybe been hearing about a new class of drugs that are being considered for myositis, the Jak inhibitors. And so those were our top active compounds. We identified the Jak inhibitors are anti-inflammatory in muscle. So those seem to be a promising kind of very soon kind of translation. As Jim said, there's already clinical trials being planned for one of these drugs, baricitinib at NIH. I know some patients are already getting Jak inhibitors off-label. So we've helped to contribute to that effort by testing all the Jak inhibitors and showing which ones are more or less potent, and kind of identifying which may be the best ones to actually use in myositis. And then kind of one step further, we've identified other drugs such as HDAC inhibitors. Not exactly important what they do, what they are, but these drugs are in clinical trials for Duchenne muscular dystrophy, which is a genetic muscle disease in young boys. And one of the promising things about that is there's now, clinical trial data in the pediatric population and in an inflammatory muscle disease. So there's something known about them. And the unexpected finding we made, which is very promising, is that HDAC inhibitors are anti-inflammatory in this interferon pathway. So those might be a good next step for clinical trials in JM. And then obviously there's all these other dozens of compounds which need to be further followed up in animal models, mouse models, and would be kind of farther down the line. The other thing I kind of wanted to point out is, so where we're going next with this program, we've graciously gotten continued funding from CureJM for another year. So I'm going to my 4th year here at NCATS. So we're going to continue doing screening, new drugs that come out. novel chemicals that can eventually be turned into drugs down the line. But we're also collaborating with a group at NCATS called Toxicology for the 21st Century. And they actually have libraries of chemicals that are found in the environment, primarily ones that, you know, humans have put out there, like pesticides, plasticizers, preservatives, food additives, things like that. So we're screening those against our assays to see, in this case, we're looking for the opposite. We're looking for potential environmental triggers of muscle inflammation. So that might be interesting if we find some kind of trigger. For example, if there's something, you know, just speculating, if there's something in your food that's causing muscle inflammation, we could then try to avoid it or regulate it or something along those lines. So we're kind of working with two halves of the equation here. Like I said, this work couldn't be done without CureJM funding to our lab, so we're really grateful for everything, and we hope to keep making progress.
You know, when COVID-19 forced us to cancel our spring and summer walks, we had to respond with a really big idea. And for the first time ever, CureJM families and friends in all 50 states have been coming together to raise support for crucial JM research. There's been an amazing groundswell of support. More than 300 families have enthusiastically joined the team, and as Jim told you, we are so excited to report that we have exceeded our goal and raised $605,000, which is totally amazing. And hopefully we've got more to come after that. So I want to acknowledge our big fundraisers who we call our heroes. Karen's going to share her screen and scroll down the list of our teams of people who have been helping already to raise money for research. You can also find this information on the CureJM website. We're truly grateful to all of our heroic supporters, and we hope that you'll join the list of fundraisers and try to raise any amount of support that you can. Every donation matters, and any help counts. Together, in just a few weeks, we have raised over $600,000, which is outstanding. I've watched every donation, large and small, add up. Every dollar we raise helps ensure our research progresses at the fastest possible pace. As a parent, that's incredibly important to me, and I'm beyond thankful for what we can do as a CureJM community. I'm also inspired and motivated to do more, and I'm super excited to announce that the CureJM Board of Directors approved a stretch goal of $750,000. And I know it sounds like a lot, but we thought 600 was a lot and we nailed it. So we feel really certain that we can hit $750,000 by July 4th. Together, we can reach this goal. I'm going to pause for just a minute so Karen can finish scrolling through the names of all the heroes. It's so incredible. The list goes on and on. So feel free to check the website. Those will be updated on a daily basis. Please know that you are a part of something big and truly important for our JM kids, as you've seen by Dr. Kinder's speech. Like my own warrior, Catherine. She is a really big warrior, as all of your children are. And without your support, we can't make scientific advancements that my daughter and your sons and daughters really need. We can do this.
So we now have only 3 days until the walk, and you're probably wondering what you can do to help CureJM reach that new stretch goal. So I have a couple tips that I wanted to share. The first one is that if you haven't had a chance yet, we're hoping that you'll take pictures showing off your CureJM t-shirt, there we go, or whatever you plan to wear on the day when you walk, whether it's before the 4th or on the 4th. Please post your Walk Strong Across America fundraiser on Facebook one more time. And if you have a chance to update the photo with your picture of you in your walk t-shirt, that would be great. Showing new photos is always a good way to get people's attention on Facebook. Or if you created a Facebook fundraiser actually on Facebook, go ahead and please share that again on your timeline. You can also send texts to friends or family who haven't donated yet, telling them that there's still time to support your walk. And we suggest including that picture of you in your t-shirt and also the link to your fundraiser page so that people can donate there. We'd like to ask you also to send photos, those photos of you in your walk t-shirts to walkstrong@curejm.org so that we can include them in our celebration video that we'll be sharing on social media and also on the Zoom call on July 5th. You can also, if you have a public social media account, you can tag your photos with #CureJM and we'll repost those photos onto the CureJM flagship social channels. So that's another way to share your photos. I really want to thank everyone for everything that you've done to make this an amazing Walk Strong Across America, our first effort for a national virtual walk. And it's been such a success so far. And I'm sure it will continue to be equally as successful over the next few days. And we can celebrate on the 5th.